everyone to IACS, International Academy of Cardiovascular Science webinar series. And it is my truly distinct pleasure and honor to welcome you all, but more importantly, welcome our distinguished speaker, Stefanie Dimmeler, who is a professor of experimental medicine and director of Institute of Cardiovascular Regeneration Center for Molecular Medicine at Goethe University. Stefanie is a world-renowned molecular biologist and eminent cardiovascular scientist who has made numerous seminal contributions to our understanding of vascular biology, cardiovascular aging, genomics of endothelial cells, and clonal hematopoiesis, and so forth. In terms of training, Dr. Dimmeler received her PhD in biological chemistry from University of Constance, but her heart was in medicine. So because of her heart was medicine, she decided to postdoctoral training in cardiovascular medicine, including in molecular cardiology at Goethe University and experimental surgery at University of Cologne. And as soon as she finished her training, she really had a rapid rise in academia, became full professor within 3 years, and became director of Institute of Cardiovascular Regeneration and professor of molecular cardiology at Goethe University. Her research I could categorize into 5 categories. Her foray into independent research started with her collaborative work with Andrew Zeyer, which was cell therapy, and here bone marrow-derived stem cells infusing into coronaries for treatment of myocardial infarction, which led a landmark article that published in New England Journal of Medicine in 2026 and has been one of the top cited articles in the field. Working on hematopoietic stem cells, he got into molecular biology and basic science understanding of it, and embarked on delineating the role of endothelial progenitor cells, endothelial biology, including genomics of endothelial cells, non-coding RNAs and microRNAs, and defining the signaling pathways that are involved in endothelial cell regeneration, angiogenesis, and in continuation of her work on myocardial infarction. And this led her to some of her work on cardiovascular or vascular aging, and including defining the vascular niches that are mediated to cardiovascular aging. And along with that, she moved on clonal hematopoiesis, continuing the trend on aging, and delineated the role of TET2 mutations as well as DNA methyltransferase 3A in clonal hematopoiesis, the role in heart failure and valvular heart disease, as well as vascular disease. Finally, the fifth column of her work is very innovative area of neurobiology in regulating cardiovascular aging. You all recall her seminal paper in 2023 defining attrition of axons in the myocardium and through a microRNA and SEMA3A contributing to cardiovascular aging. And so these 5 categories of research, which are interrelated and continuous, really a continuum of one to another, have led to over 600 plus publications, and of which 300 plus are original research articles published in major scientific journals. That has led to, I forgot the number, but over 130,000 citation according to Google Scholar, and an H-index of 184. And she is consistently, since 2004, has been one of the highly cited scientists in our time. Dr. Dimmeler, for her scientific contributions, has received numerous prestigious awards and too many of them. I counted there are 3 dozen, but I will just mention a couple of them. Hector Science Award from Hector Foundation for outstanding research achievement and a special commitment to teaching and promotion of young scientists. She also received the Otto Warburg Award as well as Gottfried Wilhelm Leibniz Prize, just to name a couple of them. She has given over 300 named endowed lectureship throughout the world and is major scientific leader both in Germany as well as internationally. She's the spokesperson for German Center for Cardiovascular Research as well as for Science for Health of the Goethe University. In addition to her academic work, she has also contributed to cardiovascular sciences through her editorship. She was the chief editor-in-chief of EMBO Molecular Medicine, associate editor currently for European Heart Journal and Nature Regenerative Medicine, as well as Science. We are most honored and delighted to have Dr. Stefanie as our distinguished speaker at the IACS webinar. Before we turn the podium to Dr. Dimmeler, I will introduce the panelists as I see them on the screen. Dr. Yvonne Deveau is the leader in cardiovascular research unit at Luxembourg Institute of Health, Department of Precision Health. I see Dr. Mahmoud Abdel-Latif on my screen. He is assistant professor of cardiovascular aging, well known for his work with Dr. Cromer. He's independent scientist now at research group leader at Department of Cardiology at Medical University of Graz. And then I see Dr. Robert Short, who is a pioneer on smooth muscle cell development and differentiation. And of course, a former collaborator with Dr. Stefanie Dimmeler in one of the first Leduc Award that they received together years ago, is professor of cell biology at University of Houston. Let's see who is my, I have next Professor Priyantosh Gerha, who is associate professor at University of Texas in Houston. Is the Welsh Foundation scholar and one of the brilliant scientists of our time. Then finally, I see Dr. Kimichi, who is well known to this audience, is the director of Center for Molecular Cardiology at the University of Zurich, Switzerland, and my distinguished colleague and program assistant, Natalie Hernandez, who is the manager of the program. With that, I'm grateful to all of you for your participation. I will turn the podium to Dr. Stefanie Dimmeler. Dr. Dimmeler, thank you.
Thank you very much for this kind introduction, Dr. Marian, and also thanks to all the distinguished panelists or discussions, which I really looking forward to discuss the science later on. It's now my pleasure to introduce what we are currently doing with respect to aging. And before starting with the research, I just would like to highlight why we are so interested in aging. So in the last years, our science and discoveries have led to the treatment of many of the classical risk factors for cardiovascular disease, including high cholesterol, hypertension. Meanwhile, also overweight, obesity can be treated by GLP agonists. However, aging remains one of the major, so far unmodifiable risk factors, and aging has a major impact both on vascular disease, atherosclerosis, subsequently myocardial infarction, but also on heart failure. And we would like to understand better what drives age-associated risk for cardiovascular disease and what are the molecular changes which are introduced by aging, making us more susceptible to cardiovascular disease. So aging is clearly associated with heart failure, which is still one of the major burdens for our health and also for our socio-economic system, with a lot of people suffering and having a poor prognosis when gaining heart failure. Age, as said, is inducing, or let's say facilitating, the introduction of atherosclerosis and leads to heart failure with reduced ejection fraction by a higher incidence of myocardial infarction. But age is also one of the major risk factors for diastolic dysfunction, which is one of the factors which lead to the syndrome which is called heart failure with preserved ejection fraction, short HFpEF. This, in contrast to heart failure with reduced ejection fraction, is characterized by, as I said, diastolic dysfunction, fibrosis, microcirculatory function, microvascular dysfunction, vascular and cardiomyocyte stiffness. While HFrEF is quite well treatable, meanwhile, there is a limited therapeutic options to treat heart failure with preserved ejection fraction, and also the age-associated risk associated with heart failure with preserved ejection fraction is currently not very clear. So we felt that there is a need to better understand what is the impact of age. And while many people, for example, David Kass very elegantly studies the effect on the cardiomyocytes, we felt that it would be important to integrate the different cell types in the understanding of age-associated pathological changes. This includes, of course, fibroblasts, which lead to fibrosis, but also the vasculature. I mentioned microcirculatory dysfunction, and also immune cell contributions, which together may change the microenvironment and thereby also progress of diastolic dysfunction. And when I talk, I will start with some of the studies we have recently done, understanding particularly impact of the vasculature and of vascular senescence. And in the second part, I will allude also to the inflammatory cells, particularly to the role of clonal hematopoiesis and mutations acquired in the bone marrow. Let me start by the question which we would like to impose: what are the changes during aging in the heart? And of course, there are many hallmarks of aging. And of course, our discussant today, Mahmoud, could tell you a lot about metabolic changes, but there are many others like genetic and instability, mitochondrial dysfunction, and proteostasis loss. And we were particularly interested as a first question, what is the role of cellular senescence and how is this occurring, particularly the different cell types of the heart? As you may know, senescence is an irreversible cell type arrest, and it has actually a positive function during development, but also in response to the repair after infarction. The short-term induction of senescence can lead to reparative signals leading to heart repair. Also, for example, liver repair is, for example, shown by these elegant studies I just cited here. But if senescence occurs chronically, as it does in aging, there is the change of the paracrine secretome, which is called senescence-associated secretome, so-called SASPs. And this can lead to a change of the microenvironment. So if you have a senescent cell, this changes the microenvironment and has influence on the other cells surrounding the senescent cells. And together with Julian Wagner, who is the wet lab scientist, and David, who did all the bioinformatic analysis, we asked the questions: when and in which cell types and which region does senescence occur firstly? So we are building currently age trajectories. So age is not a one-step process, but rather this is a chronic process leading to different types of pathophysiological changes. And today's presentation is mainly focused on a rather early time of aging, which is 18 months in mice, or which would reflect about 60 years in humans. Keep this in mind, because at later stages, you will see different signatures, and maybe I have time in 2 years or so to report to the full trajectory of aging. But when looking at 18 months in mice, we assessed the question: where and in which cell type does senescence occur at these early stages of aging? And to do so, we developed a senescence score and used spatial transcriptomics to see where the senescent hotspots can occur by taking only the cells which have the highest expression of senescence-associated genes. As said, we used known scores, but also differentially expressed genes. And you can appreciate it. This is a young heart, this is an old heart, that there is an increase in red label, meaning hotspots of senescence occurring in old versus young hearts. So with these spatial transcriptomic data sets, we can now define the regions which are enriched in these senescence score genes. And this shows you now the results in young. These are young mice hearts, these are old mice hearts. And these are the different niches which can be characterized by these spatial data. You can see also in young mice that there are some senescence hotspots, but in old mice, there's a huge increase in senescence hotspots as evidenced by this red color, which indicates the highest senescence core genes in this cluster. And this niche 7, you can then ask, what are the cellular contributions to this niche? And if you look here, this is niche 7. Red means high expression and high localization of cells to these hotspots. You can appreciate we find arterial endothelial cells, we find macrophages, we find smooth muscle cells, we find activated fibroblasts, but surprisingly, we find a relative reduction of cardiomyocytes, and also pericytes were very rare. This suggests that these early stages of aging, we find a macrovascular region, which is showing high incidences of senescent cells. And these are resembling, or let's say, comprising endothelial cells, smooth muscle cells, macrophages, and activated fibroblasts. To see whether these in silico predictions are indeed true, we assessed cellular senescence in 18-month-old mice, and you can appreciate here, this is acidic beta-gall staining. At 18 months, you see the blue staining around larger vessels. And also if we assess senescence by cell event, which is a more sensitive fluorescent staining, you can also appreciate here cell event stainings around the macrovasculature, while it was relatively rare in the interstitial region at the same time point. You can also appreciate the fibroblasts around these regions, suggesting these are really senescent regions in the macrovasculature. We also find macrophages in these niches, and when we assessed by staining CD68 macrophages, you can appreciate that the macrophage level in these adventitial regions are indeed higher and further increased in aged mice. This is the 18 months bar here in blue. Interestingly, macrophages come in different flavors, and you can separate them at least roughly by tissue-resident macrophages and bone marrow-derived macrophages. And surprisingly, the tissue-resident macrophages decline. So despite the increase in overall macrophages, this comes to the expense of a reduction of tissue-resident macrophages, and all the macrophages which are increased are bone marrow-derived macrophages, suggesting that we have a huge change in the macrophage compositions around these macrovascular niches. In addition, we assessed the gene expression in these hotspots of senescence and found that there is a change in so-called do not eat me signals. So usually macrophages can clear apoptotic or senescent cells by efferocytotic mechanisms. But if these cells express CD47, which is the do not eat me signals, and if macrophages express SIRPα alpha, they can no longer engulf these cells. So this is a block of the clearance. And you can see here in these senescence hotspots, these markers are high. You can also see this in representative immunostainings. And in addition, the macrophages showed high levels of vascular adhesion molecules, chemokines, suggesting there's really a pro-inflammatory and less clearance environment in these niches. So to sum up, I hope I've convinced you that interestingly, at early times of aging, we find senescence, particularly in perivascular niches. These niches are enriched for smooth muscle cells and activated fibroblasts, particularly bone marrow-derived macrophages, which have a high expression of so-called do not eat me signals. And I will not have time to show you this, but first preliminary data in humans also suggest that these niches also exist in humans, and we see similar senescence signatures here. This would suggest that actually vascular niches are the primary hotspots for aging, at least in these early time points. Later on, you find also the senescence in capillaries, and then at very late stages, at 22 months, also in cardiomyocytes. But that these early stages, the macrovasculature was the first hit for senescence accumulation. This actually was very similar to a study which was published simultaneously. This is a huge human proteomic study in assessing lifespan. They also find that blood vessels were the first tissue that ages. So this suggests that really there is an intrinsic effect, or let's say a high sensitivity for aging in the vasculature. If you think about that, our vasculature surrounds 2 times the world, And if you plate endothelial cells from one humans, you have a soccer field. You can appreciate that maybe this has a huge impact also on systemic aging, because if these are the first signs, we may have secondary effects on the organs, and this is something we are currently studying. What we already did study was to assess the transcriptome of endothelial cells. I mentioned that endothelial cells are a huge mass in our body, and in the heart they have crucial functions. Of course, they provide oxygen to the heart, but also, this is something which is a bit more neglected, they also export waste. They can control fibroblast activation. They are, of course, controlling immune cell invasion, and also they mediate the lymphatic system, mediate egress of the lymphatic system, of the immune cells. And parasites are controlling also endothelial cell crosstalks here. Parasites and vasculature control vessel stability. So what we asked was, what is the change in the endothelial cells early on in aging? And here we use single nuclear RNA sequencing. This is a study which was published already some years ago, comparing young and old mice. And you see here the endothelial cluster. And when we assessed endothelial cell transcriptome, we made a very surprising observation. We found actually that one of the highest GO term enriched in aged heart-derived endothelial cells were related to axon injury and neuron death. And this suggests that maybe the vasculature may interact with the nervous system. Indeed, if you go back to very old literature, we know since hundreds of years that the nerves and the vasculature co-align and co-develop. Also in the heart, you see here this nice immunostainings. Here, the pink is the smooth muscle cells. You see the nerves around the arterioles, but also around the capillaries. So there is really a tight connection. And the question was, is there an interaction between these two cell types in the aging heart? So we assessed neural density in the aging heart. You see here in young hearts and in old hearts, this is a staining of the entire heart after clearing. In green, you see the nerves. So the nerve bodies are outside, and these are the projections, the axons in the left ventricle. And you can easily see this is young, this is old, that there is a reduction in nerve density. You can appreciate here that already at 16 months, this is even before I've shown you the macrovascular niches, the innervation of the left ventricle was reduced. And we also found that this is due to the denervation of all different types of nerves. I don't have time to share with you, but we found sympathetic, parasympathetic, and sensory nerves reduced in males and females, in mice and in humans. So this suggests that there is a crosstalk between the vasculature and the nerves, leading to a denervation of the aging heart. And the question is, how is this mediated? And I included our current studies in one slide because of lack of time. What we found was that endothelial cells showed increased expression of so-called repulsive signals. These are signals which can lead to a repulsion of the axons, so a retraction of the axons from the heart. And this is one factor is semaphorin 3A. And in addition, aging reduces neuroprotective signals. So there is a disbalance of these two types of factors. So in old heart endothelial cells, we find, as said, an increase in the semaphorin 3A expression. This leads, you see here, cardiac organoids. If you add semaphorin 3A in recombinant form, there's a reduction of the nerve density. And also in vivo, if you overexpress semaphorin 3A only in endothelial cells, you can see here a denervation of the heart. And second, we found, as said, that there is a reduction of neuroprotective signals. One of these was VEGF-B. This is reduced in aging. And we could also show that we can partially rescue the age-induced denervation by overexpression of VEGF-B, suggesting that this balance here is very critical for reducing the innervation of the left ventricle. Together, this study suggests that in addition of the known hallmarks of aging in the heart, which I have already mentioned, we also have to add denervation to the critical changes in the aging heart. And of course, it will be very important to study how this impacts then cardiac pathology and age-associated dysfunction. We were wondering what may trigger semaphorin 3A and also these changes in these neuroguidance molecules. And interestingly, coming back to the senescence story, we found that semaphorin A was increased in the supernatant of aged endothelial cells, suggesting that semaphorin 3A may be part of the senescence-associated secretome. This led us to hypothesize that maybe senescence could be an upstream regulator for denervation. So we asked whether senescence, or at least the senescence-associated secretome, may contribute to denervation. To test this hypothesis, we used senolytics. These are drugs which can selectively kill senescent cell. This is quite long known. There are different types of molecules which you can use to induce the cell death of these senescent cells. And we used a classical senolytic, which is dasatinib and quercetin, which is also applied in early clinical phases, for example, in Alzheimer's disease. When applying these substances to aged mice, this is now a study where we compared aged mice plus minus placebo or senolytics. And then after 2 months of treatment, we assessed, first of all, senescence. You see here, these are now mice which are 2 months older. You see also senescence not only in the macrovascular niches, but also in the microvasculature. And if you treat the mice with senolytics, you see clearly a reduction of the senescence in these hearts. Simultaneously, we find an increase in axon density. You see here the quantification on the right, suggesting that indeed the clearance of the senescent cells can rescue the innervation of the myocardium. What is the consequence function? So we assessed some of the features which could be influenced by nerves, and one is, of course, heart rate variability. We know that elderly have a reduced heart rate variability, which is associated with a poor prognosis. And you can appreciate that the heart variability was increased by senolytic treatment in these old mice. Interestingly, also the circadian rhythm was restored. So you see here the day-to-night cycle in old mice in red and in young mice in gray. And senolytics tend to restore the heart variability in a day-night rhythm. Second feature of nerves are, of course, electrical stability. It's known that denervation can lead to silent ischemia and lethal arrhythmias. And indeed, we could show that senolytic treatment partially restored the higher incidence of arrhythmias in these old mice-derived hearts. Finally, one of the key hallmarks of aging is, of course, diastolic dysfunction. And we assessed diastolic dysfunction and fibrosis as well, and could also see in echocardiography and graphic analysis that the senolytic treatment, see here in this light blue, compared to the placebo treatment, reduced diastolic dysfunction and also reduced to some extent the fibrosis. This suggests that during aging, we have a change in the axon density, a change in the senescence, particularly of the vasculature, which leads to a disbalance of these factors which control axon regression. At the same time, we also find a change in the macrophage populations around the vascular niches, a disbalance. I think this is also known by others, that the bone marrow-derived macrophages are increasing while the tissue-resident macrophages are reduced, suggesting that there is a strong interaction in these regions between inflammation and also the neural system. And I think currently we are also trying to understand how these neurovascular immune crosstalks in aging can contribute to this disbalance, and maybe also how the lack of clearance mechanisms, which are impaired possibly during aging, may contribute to this acceleration of senescence, accumulation of senescent cells, and subsequent aging. Of course, this all looks very promising, but of course one has to acknowledge the limitations of senolytic treatments. You have to find the right spots. If you have too little senescent cells, you will not have any effect. If you have too many senescent cells, this likely also is detrimental, because if you kill a lot of cells, you can appreciate that this likely leads to inflammation and tissue destruction. So there might be a need to find the sweet spots for senolytic treatment early enough to have also repair capacity, because the tissue after the killing of the senescent cells also needs to repair. So this may limit the senolytic treatment to relatively early stages of aging. And there may be a need, and this is what we are currently doing, to develop biomarker-guided therapies to also get hints on biomarkers which may tell us about senescent cells in the heart and these early types of maybe patients or healthy aging people who may need senolytic treatment. And also, of course, there's a need of alternative strategies. And one thing which we are currently explored in the last months and last years was how the epigenetic control can change the aging of endothelial cells. So we assessed how epigenetic control of endothelial cells may be regulated during aging, and we did so by performing ATAC sequencing with single cells. And we found that in endothelial cells of the aging heart, one of the highest repressed transcription factor was zinc finger and BDB domain containing protein 16. Actually, this chromatin around the locus for this gene was very strongly repressed. And we found that this gene was repressed both in human and mouse endothelial cells in aged hearts. So you can see here a volcano plot for single cell data. You can even see this on protein expression in Western blots from mice. And of course, we asked, what is the role of this transcription factor? Could it be that this is a key regulator of aging in endothelial cells? And to get first hints, we used heterozygote mice. So homozygous mice have a strong pathology. They also have changes in the skeletal development. But heterozygote mice don't have obvious Mendelian ratios and obvious phenotypes. But if we let them age to the 3 months age here, we find that despite only a half maximal repression of ZBTB16 endothelial cells, there is a significant induction of diastolic dysfunction, increase in left ventricular mass. There's no systolic dysfunction, but cardiomyocyte size and also the senescence, particularly again around these vascular niches, was significant augmented, suggesting that this is indeed a critical regulator of aging of the heart. Of course, this is a global knockout. This transcription factor is also expressed in other cells. To see whether this is really endothelial driven, we generated endothelial-specific knockout mice. And I don't show you all the data because I don't have enough time, but you can appreciate the diastolic dysfunction at 4 and 8 weeks after specific induction of the deletion of this transcription factor. We also find left ventricular mass and hypertrophy and vascular senescence, suggesting that endothelial cells can be the driver of this diastolic dysfunction and aging heart phenotype. What could be the mechanism? We performed, of course, RNA sequencing and found a lot of genes which have paracrine functions again after ZBTB16 silencing to see whether the paracrine effect of these cells could have an effect. We generated conditioned media from endothelial cells silenced with Z4-ZBTB16 and then used the conditioned medium to treat, for example, fibroblasts. And we found that this conditioned medium indeed induces fibroblast activation, collagen 1A1 expression, smooth muscle actin expression, contraction, and fibrosis of cardiac organoids. One example is shown here, collagen 1A1 expression in the ZBTB16 supernatant treated fibroblasts, or here an organoid model. You see here smooth muscle actin and collagen expression if you treat the organoids, the cardiac organoids with these supernatants. Also on cardiomyocytes, we found differences, and we see here an example of a co-culture system. So this is not the conditioned medium, but rather the co-culture of silenced endothelial cells. You see that the endothelial cells are also not entirely happy, but the cardiomyocytes clearly show an increase in hypertrophy under this condition. We also measured a change in beating frequency, which is often a stress response, and found that there is also an impaired crosstalk between endothelial cells and cardiomyocytes in this context in vitro. Finally, we of course looked into the denervation, and consistent with our data suggesting that endothelial cell senescence and aging can lead to a denervation, we also find here in this supernatant treatment a reduction of nerve density, and also we could see this in vivo in our mice models, suggesting that this transcription factor in endothelial cells, which is suppressed during aging, has really serious impacts on other cells of the heart, and this may contribute to the dysfunction. Of course, to see whether we can find a therapeutic approach based on this data, we finally assessed whether a gene therapy approach could work. And to do so, we used AAVs overexpressing ZBTB16. This was done in collaboration with the group of Oliver Müller and also Manfred Okrist from Vienna and Christian Kuppert. They developed a system which really works very nicely in endothelial cells with a panam-conjugated peptide, which conjugates the virus and makes them more tropic for endothelial cells. And we used an endothelial promoter to then drive endothelial cell expression. You can see nicely that this is really working in the endothelial fraction. Also, we could observe already after this relatively short time treatments for 2 months with these vectors a reduction in diastolic dysfunction. And we could at least prevent the further increase in fibrosis. You can see here 18 months we started the treatment, and this really prevented the further induction of fibrosis, but it didn't bring it back. So it's not a reversion of fibrosis, but rather an inhibition of further subsequent fibroblast activation. With this, I would like to conclude this part. I hope I've convinced you that this chronic senescence indeed can be detrimental. Altered epigenetic control of gene expression, like the repression of ZBTB16, can drive endothelial cell senescence, and this then leads to an altered paracrine activity, which leads to fibrosis and also cardiomyocyte dysfunction. And of course, with this, I did leave out so far the inflammatory cells, but of course, as said, there is a close crosstalk between inflammation, vascular aging, and diastolic dysfunction and also cardiac dysfunction. So in the last couple of minutes, I would like to tease at least the concept that this all is aggravated by bone marrow-derived cells, which can comprise mutations acquired during lifespan, which can lead to clonal hematopoiesis. And actually, these mutations accumulate in our body throughout our life. But the hematopoietic stem cells are particularly, I would say, sensitive because they have to renew so often that we can, after acquiring these mutations, they can have a survival benefit. And the mutation of some of the genes, for example, epigenetic regulators like TET2 or DNMT3A or other signaling molecules, can lead to the expansion of these mutant blood cell clones. And they can have effects then on the heart. In addition to these mutations in driver genes, one chromosome alteration, which is the most often cause actually for clonal hematopoiesis, is the loss of the Y chromosome. So males, which are above 70 years, 40% of them have a loss of the Y chromosome in their hematopoietic compartment. And this both together has very strong impacts, we believe, on age-associated changes in the heart and also, of course, the vasculature. You see here that both the chip driver mutations and the loss of the Y chromosome is strictly age-dependent. So it, of course, can aggravate age-associated cardiovascular dysfunction. And this is now only shown for the chip driver mutations, but they have a strong impact on the worsening of cardiovascular disease, including atherosclerosis, but also heart failure, HFpEF and HFrEF actually, and also aortic valve stenosis, stroke, and lung disease. So this is just one example for heart failure. I could show many data by many very important discoveries from the literature. Here you can see that the incidence of these mutations is higher in disease. In addition to age, disease also aggravates the occurrence of these mutations. This is, for example, heart failure compared to a control cohort. And also, if you have these mutations, this is our heart failure cohort, the patients have a poor survival and prognosis in comparison to patients which have no chip driver mutations. Also, in mice models, this has been substantiated. So it's not only a bystander, but at least in mice models, if we transplant mutated cells or cells having a half maximal or inhibited expression of these driver genes, this also can lead to the aggravation of, as said, atherosclerosis and heart failure. preserved ejection fraction and reduced ejection fraction, suggesting that this has a very important contribution. We were investigating actually what is the direct role of these mutations in the blood cells in humans, because most of the studies at that time had been assessing their effect in mice models. So we did single-cell sequencing of no-chip subsets, patients and DNMT3A and TET2 mutations. And you can see here that inflammatory cytokines such as interleukin-6, the interleukin-6 receptor, interleukin-1 beta, macrophage activation, and NLRP3 were increased in expression. So these are single-cell data, and these Volcano plots, you can see this increased expression. And also, if you look in the Go term, there's a clear evidence for cytokine production, macrophage activation, and a chronic inflammatory response. We also nailed this down to the specific mutated cells by using an advanced single-cell technology. If you want to look this up, it's published in Nature Cardiovascular Research recently, and documenting that the mutation itself in humans can lead to change of the gene expression profile. To get some hints how this may interfere with the myocardium, we integrated our data sets and found actually that in addition to the inflammatory signals which are provided, there is also a strong crosstalk to fibroblasts. And this led us to hypothesize that maybe a paracrine activation of cardiac fibroblasts can be also induced by the mutant blood cells. And to mimic this, we silenced DNMT3A in macrophage populations in vitro and assessed the supernatant and could see that indeed fibroblasts were activated both in fibroblast cultures as well as in cardiac organoids. We also found an increased fibrosis in mice where we mimicked the human DNMT3A chip driver mutation and an increase in fibrosis in the remote zone. And finally, we also assessed this in MRI studies using T1 imaging together with Eike Nagel. We found that patients with CHIP have a higher incidence of cardiac fibrosis, suggesting that these CHIP mutations have an effect on inflammation as well as on possibly also fibroblast activation. So to sum up, we believe that the bone marrow-derived compartment can aggravate this cardiovascular disease, and particularly heart failure during aging. We have an increase in bone marrow-derived macrophages in the aging heart, which then, of course, can lead to recruitment of these CHIP mutations carrying monocytes, leading to the then differentiating to macrophages. They have a clear pro-inflammatory signature, but also some of them, for example, DNMT3A mutations, can lead to fibroblast activation. I didn't have time to show you so much, but I find it very exciting that the loss of the Y chromosome, which is, as said, very high prevalent chromosome alterations in men, has a very strong effect on macrophages. And we found also in humans, I don't have time to show you, that there is a paracrine fibroblast activation induced by these macrophages. And this is really very strongly fitting to what Ken Walsh showed very elegantly in his initial science paper, where he discovered that loss of Y leads to cardiac fibrosis, and additional studies in humans where we recently showed all the signature in the UK Biobank, which resembles this fibroblastic activity of this loss of Y mutant carriers. With this, coming back to the original findings, of course, we have to do some housework now, because the hypothesis based on this data would be that CHIP or loss of Y may also aggravate this senescent vascular niches. So we speculate that maybe these CHIP driver mutations may lead to an aggravation of senescence and the activation of fibroblasts in these vascular niches, because macrophages will accumulate there. And this is very strongly fitting also to data from Ken Walsh. He showed in the kidney that also loss of Y macrophages can have a defective clearance function, so they may lead to the accumulation of senescent cells, at least in the kidney. With this, I would say a bit speculation about the interaction between clonal hematopoiesis and cardiac senescence and the vascular niches. I would like to end and would like to point out that with the diagnosis of CHIP or loss of Y, we may have a chance also to select high-risk patients together with our biomarkers for aging. We may find subjects where we need an early preventive strategy with anti-inflammatory, anti-fibrotic, or possibly anti-senescence, anti-aging drugs. With this, I would like to thank the people who did the work. I would like to thank all the group and also funding agencies. For the first part, I mentioned Julian Wagner. He's really the driving force of the aging studies I have shown you together with David Rodriguez Morales, but also others in the group and many collaborators. And for the chronic hematopoiesis study, I would like to particularly thank Mariana, who did this paracrine activation study with DNMT3A mutant cells I've shown you, and Andreas Zeyer, together with Timo Speer, who did the clinical studies behind them, and our bioinformatic team with Wesley and David John, and of course, all other contributors. With this, I would like to stop sharing my screen, and I'm happy to address any open questions.
Thank you, Stefanie, for this magnificent presentation. It's a beautiful, comprehensive data on various aspects of vascular aging. Is fantastic, and we are very grateful to you for this beautiful presentation. With that, I think we open the presentation to discussion by the panelists and followed by attendees if they have any questions. Let's start with the panelists. I see Dr. Robert Schwartz. And I'm getting here. Screen is moving too fast. Too many people raising hand to discuss. I think Robert Schwartz was the first. No, was I mistaken?
Yes. Yeah. It appeared and disappeared so quickly. I haven't talked to Stefanie in years. We shared a major grant many years ago. It's delightful to hear your work and how far you have gone. It's on a level of spectacular. I don't know if you know any of our stuff that we've done recently in 2022, but we show two mutant transcription factors can repair damaged hearts. One was called STEMIN and one was called YAP5SA. And this was published in Cardiovascular Aging. And the remarkable discovery was that these two mutant transcription factors activates OKSM, activates the Yamanaka stem cell factors. And we felt that that was also close to the data that Thomas Braun showed, that stem cell factors are reparative. And we couldn't figure out why this was all so powerful. And we just have a paper now in press in iScience. And what it shows is that these two factors stimulate exosome secretion of anti-apoptotic microRNA, and some of the microRNA is also anti-inflammatory. So if you go back and look at our figures in the second paper in regeneration of the heart, you see virtually no fibrosis of the heart. And we have now a pig study going on in Louisville, Kentucky, which we believe is showing similar results, improved cardiac function with injection with this RNA post a major infarct, and also increase in cardiac mass. This was actually a question that was raised by A.J. Marion when he was an editor of Cardiovascular Aging, and asked me, how can this all be a result of replication? And what we discovered is probably not all replication. It's the increase in small microRNA, over 100 microRNA, both induced by STEMIN and YAP mutants. Parts of them are secreted in exosomes, and they block cell death, and they block staurosporine cell death. So the question is that this might be a paracrine autocrine event that helps reduce a lot of the inflammation and death. The other thing we found that I think impacts what you look at in monocyte infiltration was that we saw an increase in angiopoietin-like 4, which has been shown to be involved with polarization of monocytes. And that may also reduce the infiltration of monocytes into the heart. So although I believe what you found is seminal in your regulatory factors, there's more going on in the inactivated chromatin that may also be adding to your incredible observations.
Thanks, Bob, so much for sharing also your thoughts and the new data with us. So I certainly agree. I didn't have time to show you, but there is indeed a very surprising upstream regulator microRNA. I have to look now in your paper to find out whether this is the shared ones.
I'll one's in press, so you won't be able to see it immediately, but I'll send it to you.
Yeah, I will watch out. So what he found is if we knock out this microRNA, there's really, we see this age and we see changes in fibrosis. So there is really profound changes as well. And I think that it's, of course, more complex. We did not yet figure out what are the paracrine factors, actually. It can be exosomes, it can be also metabolites. So I think the crosstalk will be not unidimensional. But if you have powerful transcription factors which affect many aspects, then, of course, you may have a really instrument in your hands to change the paracrine environment and thereby also affect aging. So I'm very much looking forward to the upcoming paper.
Thank you.
Excellent. I see on the screen in order of the, I'm not sure what order, I lost it, forgive me, but from the screen to the right, Dr. Gerha.
Thank you very much. That was a very fascinating talk. So from your talk, it looks like somehow the cardiac cells are pretty resistant to senescence. If we look at monocytes, fibroblasts, and it seems to be a very surprising finding. Whereas the macrophages and other cells, maybe because of the chip mutations or something else. So how do you explain this sort of resistance in cardiac cells to senescence? And is it just because there is a slow turnover of senescence that at a given time point, maybe you may find a lot more macrophages undergoing senescence, but the cardiac cells are very low rate and you're not able to really detect them? Or is this something very unique to the chromatin state of those cells that actually prevent the senescent phenotype?
This is an excellent question. If you would have asked me before, I would have said endothelial cells and cardiomyocytes are the first. But the data are the data, so it's spatial transcriptomics, it's single nuclear sequencing, it's immunostaining for different markers. I know senescence is not an easy task to predict or to show, but what we have done points to a critical change early in these macrovascular niches, and then endothelial cells, and then cardiomyocytes. Maybe the cardiomyocytes are a bit more stress resistant because they do not proliferate. This could be one feature, but I also don't believe that telomere erosion in endothelial cells would lead to the senescence. So I don't believe that replicative senescence is the major driver. So I think what we currently do is we try to look in the different repair capacities and the genetic stability of the different cell types, and there are huge differences. But of course, these are also a lot of different genes and gene classes, so I can't tell you yet. And problem is also smooth muscle cells seem to be very high senescence very early, and they are a very tiny population of cells. So we have to enrich them. And then maybe I can give you more hints. One idea I have, just the last word, is that maybe the macrovascular niches are also earlier stiff or earlier stressed, that the region also is somehow contributing to these stress responses, like piezo's or whatever. This is something, but I can't give you an answer. I can't just speculate. It's an excellent question. We hope we can solve this in the future.
Great. I have Dr. Giovanni Camici next on the screen.
Yes, thank you. Thank you, Stefanie. Really incredible how much you managed to put together in just 40 minutes. Really very, very interesting. So I have a question concerning the last part of your talk, how you explained to us how loss of Y and CHIP exacerbate aging process and also inflammaging. And I was wondering, of course, it's extremely important to understand mechanisms, how this takes place, how different cells interact, but it's equally important to identify targets and mediators so that we can then eventually design therapeutic strategies. I saw among your mediators, you had some of the classical cytokines, IL-6, IL-1 beta, and I was wondering, have you, or if in case you didn't, do you think it would be possible to repurpose some of the currently clinically assessed antibodies, like for instance, ziltivekimab and others in this specific setting? I mean, I think it would be very interesting to know whether you've done it or whether you think it would be a good idea.
So certainly this is the way to go. So there are studies with low-dose colchicine because this is cheap and you can do this on an academic level. So Moritz van Scheidt has finalized the study, and also I think and is having study, and we also have a study in heart failure with low-dose colchicine in CHIP patients to see whether proof of concept surrogate markers are changed. But I think we have a grant application for anti-interleukin-1 beta in TET2, and we would love to test anti-fibrotic therapeutics in DNMT3A carrier. So we believe that DNMT3A in heart failure has a more pro-fibrotic effect than maybe finerenone or something like this would be a good way to go in this subset of patients. So yeah, I'm not a clinician, so I can just give my ideas and try to push the protocols, but I hope that we can get some things to start. And it's of course very expensive because industry wouldn't like to limit the patient population so easily. Yeah.
Thank you very much.
I have Dr. Mahmoud Abdel Latif next on the screen.
Thank you, Marion. Stefanie, as usual, fantastic talk, really. It was really a pleasure to listen to your presentation and all the data you shared. And I had a very specific question about the ZP-Z16 silencing and whether it affects neighboring cells, in the sense when you knock it down or silence it in endothelial cells, particularly in a cell culture system or this using the conditioned media. You showed elegantly how you can modify hypertrophy in cardiomyocytes or activation of fibroblasts. Have you checked whether also senescence levels in those cells are affected or not? This is
What? Sorry.
whether senescence levels in those activated fibroblasts or hypertrophied cardiomyocytes, does this also play a role in this process? I don't know if I missed it.
Yeah.
Have you checked?
We find that if we silence ZP-TB16 in endothelial cells longer, we find senescence of endothelial cells. But we did not find, to the best of my knowledge, senescence of cardiomyocytes or fibroblasts. So, but these were short-time cultures, so I can't say whether this.
Yeah, I'm not sure if it's even technically feasible to do that, but I'm asking this because I remember the late Judy Campisi always said there are no matter how many senescent cells we detect, in absolute numbers, there will always be a very small proportion. And this brings me to the more hypothetical question that I wanted to ask you. If all senescent cells are the same, maybe very few senescent cells are so much worse than many in a different cell type or a different life stage or a different disease. Just what you think about this.
So I agree that the senescent cells are, if you would make a percentage in the heart, are not so high. But if you look on these vascular regions, I would say they are quite significant, yeah. So locally they are accumulating. This was very surprising to us. And then of course they can have a very strong local influence. Also, I would say if you think about how, if you have a senescent capillary, this is quite long in the it's not, if you make a cross section, this may seem very little, but the interaction with cardiomyocytes is not so little, the phase they need. And the paracrine effect with exosomes or other paracrine metabolites, they can have significant effects. Of course, this is now, we would need very sophisticated models to look specifically knock out the gene than in senescent cells, and this takes with 18 months it takes ages, and therefore we just knock the gene out in all cells, and this of course is not so elegant as you would like to see to pinpoint what is the specific role of the senescent cells.
It is elegant, but it's just we're always looking at the next step. What can we do better?
All right, thank you. The next I have on the screen, Dr. Yvonne Deveau, please.
Thank you very much. Thank you, Stefanie, for this very inspiring lecture. I learned a lot, which is a good sign. My question is related to biomarkers and to the fact that you nicely show that if senescence is already too far away, then we cannot really go back, and it's difficult to reverse, but maybe eventually to prevent. So in terms of biomarkers, do you more focus on diagnostic biomarkers, meaning biomarkers that would indicate that senescence is already there, or is there any possibility to predict whether senescence cure? For example, if we think of a heart attack, after myocardial infarction, you know that a certain percentage of patients, they would develop ventricular remodeling. And it would be very interesting to see if some markers of cellular senescence could be detected at an early stage after the myocardial infarction. And then we could act and try to prevent the remodeling process, if it's the case. Do you have any data related to that?
So we and also others have data with respect to senescence after acute myocardial infarction. It's very interesting. There is an early boost of senescence in the first days. This is mainly in fibroblasts. This has been shown by Stefan Engelhardt's group also. And then later at 1 month and 2 months, where the chronic remodeling occurs, then we find also endothelial cell senescence and the macrovascular niche senescence type, which we also see in the aging heart. So there is really a biphasic occurrence of senescent cells, and we are currently testing ZBTB16 or also these other mediators of senescence in late stages after myocardial infarction. So I don't believe that we should treat these early phases, because they may be reparative, and also fibroblast senescence may be something good, but the chronic phase where we have this remodeling, maybe this is really detrimental, and we currently find out. With respect to the biomarkers, there are very good groups like Tony Viscorine. They try to find biomarkers which are organ-specific, and very disappointingly, the only aging marker they found for the heart is anti-probenpin and troponin, which we would say that certainly this is not specific enough for detecting aging in this. So I also didn't find very convincing vascular biomarkers for aging. So I think there needs more work to be done with this respect to hopefully get some signatures. I think it would be really elegant to measure the burden of senescence, particularly of cardiovascular senescence, if we can, or combine it with the different organ senescence and then find patients which would, or maybe they are not yet patients, preventive strategy for those. And my vision would be not to use chronic treatment, but rather use like a short-term diet, You give senolytics for a few weeks. We are testing now short-term protocols, and then you clean, and then you block this vicious cycle, and then you can do this maybe in a few years again, but that you can get a break of the further accumulation of this process. And this is what we are currently testing. We will see how this evolves, at least in the adipose tissue. Some studies showed that short-term treatment also can be useful, and this would be, of course, reduce also the risk for the patients, because like a short-term period would not have such a problem with side effects, which we cannot tolerate in a relatively healthy population of subjects.
Absolutely. Thank you very much. If I may, because I did not lower my hand, what about sex differences? You didn't really talk too much about sex differences in your presentation. Do you know whether the cellular senescence pathways are different, and could explain, at least in part, why women have a longer life expectancy than males?
So there are two things with respect to sex differences. So one is that we see more profound effects in females, both with respect to denervation and to ZBTB16. Effects are stronger in females. So there seems to be a higher sensitivity for females, particularly in humans. So in our human heart sections, the denervation occurs very early, much more profoundly than in males. And the second thing is, of course, the loss of the Y chromosome. I think this is something which, strangely, the loss of the Y, if you look in our clinical studies, if you remove the loss of the Y patients from the male cohort, they have an equal risk as the females. So funny enough, the loss of the Y may really contribute strongly to the higher risk of cardiovascular disease in males. You can look up the data. It's maybe limited to our cohorts, but there is a strong signature which we see in this respect as well.
Great.
And the Y chromosome is very sensitive. It's very small. It can be easily lost. So disadvantage for males. The X chromosome, the loss of the X chromosome is not as often.
Excellent. So we have two questions from the audience. Dr. Francesca Vicente would like to know, thank you for the amazing talk, Dr. Dimmeler. This is Francesca Vicente. I was wondering whether you had the chance to observe any changes on the coronary flow reserve velocity and microvascular dysfunction in your ZBTB16 mice.
This is an excellent question. We didn't use this. We just measured the capillaries morphologically, which is a clear limitation. So we see effects on the diameter of the capillaries and also reduction in numbers. But unfortunately, we did not measure it because the laser Doppler in the heart is not so easy. Yeah.
Very good. We have one more question from Dr. Frederica Morelli-Berg. Dr. Berg would like to know, great talk, thank you. Do you find T cells in the senescence perivascular niche? If so, what do they look like?
We need further data for this, but we definitely find T cells.
Yeah.
More to come, but T cells are complicated, so we need more power and more data for try to dissect the different types of T cells.
With that, I'd like to thank you, Dr. Stefanie Dimmeler, for your fantastic host this IACS webinar, and this will be at the web page of IACS, and will be available in a couple of days. Thank you again, Dr. Dimmeler. Our sincere gratitude to you. Thank you, panelists, all of